Assalamualaikum and a very good morning. A warm welcome to all participants from Malaysia, the SEAMEO network and affiliated countries to IMR-SEAMEO Webinar Series 2023. The theme today is Uncover the Hidden Diagnosis of Rare Genetic Diseases. Our presenters today are from the Institute for Medical Research in Malaysia. I am Izzati Yaakob, the head of Molecular Diagnostics Unit, Specialized Diagnostic Center, IMR, will be the moderator for today's session. Before we start, I would like to take this opportunity to announce our activities currently held at the National Institute of Health Malaysia. The Institute for Medical Research, in collaboration with the Persatuan Penyelidik KKM, are organising Rare Disease Awareness Week 2023 or Minggu Kesedaran Penyakit Jarang Jumpa 2023 in conjunction with the celebration of World Rare Disease Day 2023 on 28 February this year. The purpose of organising this event is to raise awareness among National Institute of Health community about rare diseases with various exciting activities and programs that have been planned throughout this week. Until next Tuesday, for the latest information, you can visit the official Facebook page of Minggu Kesedaran Penyakit Jarang Jumpa MKPJJ NIH 2023. Ladies and gentlemen, this webinar series was initiated with the aim in mind to share the knowledge, discuss topics on biomedical research, and to present research outcomes. Before we begin our presentation, allow me to announce a few housekeeping messages from the Secretariat. We will not be taking questions during the presentation, but participants may post their questions using the link shared in the YouTube channel. The question and answer sessions will be conducted after both presentations have ended. The attendance link is also provided to those who would like to receive an e-certificate of attendance. You may find the link in the chat box of the YouTube channel. Last but not least, please subscribe to our National Institute of Health Malaysia YouTube channel and click for notifications whenever there is a new post. Ladies and gentlemen, for today's webinar, we have two speakers. Our first speaker is Dr. Nurul Izzati Hamzan, who is the clinical scientist in Special Protein Unit, Specialized Diagnostic Center, IMR. She holds a doctoral degree in microbiology from the University of Science Malaysia, USM Kubang Kerian, Kelantan. Her research area of interest is on microbiology, molecular and protein tests. Currently, she is working on project for establishment of multi-TOF mass spectrometry for analysis of congenital disorder of glycosylation in pediatric samples. She has presented and published many papers in national and international journals. For today, Dr. Nurul Izzati will be sharing her research experience with the presentation entitled Congenital Disorder of Glycosylation or CDG Diagnosis, Past, Present and Future. Dr. Izzati, the screen is yours.
Thank you, Puan Yusnita, our moderator for today, for a very kind introduction. My presentation today is entitled Congenital Disorder of Glycosylation, CDG Diagnosis, Past, Present and Future. I am Nurul Izzati Hamzan, a clinical scientist in microbiology from Special Protein Unit, STC, IMR. So the presentation today will cover an overview of CDG, which included protein glycosylation, type of CDG, clinical presentation, and also the prevalence. Secondly, I will discuss on the laboratory technique, the past, present, and future in diagnosis of CDG, and what are the direction in IMR. Okay, as a brief introduction, CDG, based on the infographic by World CDG Organization 2016, CDG is an umbrella term for a rapidly expanding group of rare metabolic disorder due to the genetic defect in complex biochemical process known as glycosylation. And the glycosylation itself is the process by which glycan are covalently attached to protein or lipids, forming glycoprotein and glycolipid respectively. So the glycosylation process occurs either in the endoplasmic reticulum or in the Golgi system. And based on the current guideline, CDG is classified into these 4 glycosylation defects. But the most common are due to the N and also O glycosylation defects. Okay, why glycosylation is very important in CDG? This is because proper glycosylation here will lead to a healthy child while improper glycosylation will lead to child with CDG. And in diagnosis of CDG, transferrin molecule is very important. Why? Because it the most abundant glycoprotein in human plasma. It is also a widely used rapid biomarker for detection of the end glycosylation defects. Okay, in normal condition, transferrin has 4 salicylic acid here and 2 glycosylation site. And in CDG type 1, they are missing one or both glycosylation site here, and this occur in the endoplasmic reticulum. For CDG type 2, as you can see, there are modification here in the already transferred glycan. It is either occur at one or both glycosylation site, and this one occur in the Golgi system. Okay, diagnosis of CDG is often challenging because of the clinical presentation are extremely heterogeneous. Majority of CDG with end glycosylation defect has multi-organ disease with neurological defects. The classical features, clinical features of CDG are as displayed. They have dysmorphic feature, facial features, intellectual disability, inverted nipples, and development delay, and also epilepsy. And over the year, up to date, there are over 150 CDG type has been described. The prevalence of CDG. The prevalence of CDG specifically PMM2 CDG in Dutch population is 1 in 77,000 people, and Polish population was estimated at approximately 1 per million. CDG in Saudi population is 11.5 per 10 million people. And in Malaysia, the reported CDG cases was published by Tong et al. On year 2009. The case was identified in Malaysia in a female at 2 days of life, and the diagnosis was suspected on the basis of abnormality of serum manifestation and was supported by abnormal serum transferrin isoform pattern. Current data from Malaysia that we get from the Genetic and Metabolic Clinic HKL, it showed that we have up till today only 13 patients with the confirmed cases of CDG. Okay, this is how the diagnostic algorithm for CDG. First line screening is by isoelectric focusing of serum transferrin. Then, based on the pattern of the transferrin, we can classify into abnormal and normal. The abnormal one, we are further classified into type 1 and type 2. For primary type 1 CDG, further classification is done by enzyme and mutation analysis, while primary type 2 will be classified based on glycan structure analysis. Okay, for primary type 2, we are further classified using isoelectric focusing of apolipoprotein C3 for the diagnosis. And the confirmation is only can be done by using targeted sequencing and also next generation sequencing. Okay, this is the timeline for laboratory diagnosis of CDG in IMR. IMR has started the test on year 2005 by utilizing IEF, isoelectric focusing of transferrin isoform. And this method was developed by Puan Norsia and Dr. Nurul at Unit Molecular Diagnostic and Protein. And today and from 2018 till today, we are changing the method from IEF to the capillary zone electrophoresis CZE. And for the future analysis, IMR aim to embark on the Marditov mass spectrometry, where I will discuss on this method on the later slide. In the past, for IEF fast system machine has been used, and this is how fast system machine look like. The principle of fast machine is that fast system is analyzing the pattern of glycosylation of transferrin based on the isoelectric focusing. molecules will be separated based on their isoelectric point, and transferring isoform are visualized by staining the gel with the protein-specific dye. For the present technique, we are using CZE, and this is the analyzer used and placed at our unit. It is an analyzer CBR capillary 2 flat piercing and was installed in July 2018. The principle for CZE is that capillary electrophoresis is a liquid-based separation technique that uses a capillary as a separation channel under the influence of electric field. It is an analytical technique where the electro-osmotic flow helps separate charged ion. So the result interpretation is based by viewing the position of the separated isoform fraction captured on the specific software and can be viewed in the computer. As you can see the picture here, this is the separated isoform fraction. Okay, to conclude, to conclude and to compare the past and the present way of diagnosis CDG, IEF is a manual process. It is a gel-based method. It is laborious, but it requires only low sample volume. For capillary zone electrophoresis, it is automated process. It is software and computer-based, so less training required to run the test, but it requires high sample volume. Using IEF, the band pattern will show type 1 CDG, type 2 CDG, mixed pattern, and also polymorphism. Using CZE, the silo pattern will show type 1, type 2, mixed pattern, polymorphism, interference, and also complement. Okay, the drawback of this technique is that it cannot provide subtype information on CDG, and the presence of polymorphism, interference, complement will alter the isoelectric point in the IEF and modify the silo transferring profile in CZE. And this polymorphism, interference, and complement does not cause dizzy. Hence, it will hinder the interpretation and the diagnosis of CDG. Additional reagent and buffer required to discriminate between this pattern. It is more time consuming, and of course it will be more costly. So, what are the future direction for CDG at IMR? At IMR, currently, IMR currently conducted a study on establishment of Mardi-Topf mass spectrometry for CDG in pediatric sample, and this will be completed on 2025. So what is Mardi-Topf? Mardi-Topf stands for Metric Assisted Laser Desorption Ionization Time of Flight Mass Spectrometry. The principle is that it is an analytical technique in which particles are ionized, separated according to their mass to charge ratio, and measured by the time it takes for the ion to travel to a detector at the end of a time of flight tube. Why we choose to embark in Mardi-Topf? In CDG diagnosis, Mardi-Topf is used to analyze the pattern of the N-glycan. And why glycan is very important? Because it has several advantages to study glycan in CDG. Number one is can be used as a diagnostic tool. Glycan analysis can provide useful diagnostic tool for identifying and characterizing CDG subtypes. Number two, the glycan analysis can provide underlying mechanism of CDG. And as for biomarker, glycan analysis can also become a biomarker. It is a very sensitive and specific biomarker for CDG. Okay, the advantages using Mardi-Topf itself, the method itself for detection of the CDG is that it has high throughput. It can analyze multiple samples in short amount of time. It is comprehensive. As I said before, the pattern of N-glycan will allow for more accurate diagnosis and subtype classification. It is more cost effective compared to other analytical methods such as genetic testing or enzyme activity assay. Moreover, it has rapid turnaround time, which allow for a more rapid diagnosis of CDG, and it has low requirement of sample volume. So, as a conclusion, CDG in Malaysia are underdiagnosed, possibly due to the lack of availability of sensitive and specific laboratory testing. Therefore, IMR aim to expand the laboratory diagnostic tests offered for screening and diagnosis of CDG in order to aid clinicians in detecting CDG among suspected patients. As for the acknowledgement, I would like to thank Special Protein Unit SDC, and this is my team members for CDG, and that's all from me. Thank you. Okay, now I pass back to the moderator.
Thank you, Dr. Izzati, for the interesting talk. If there are any questions that you would like to ask about her presentation, kindly submit your questions using the link provided in the YouTube channel. Next, I would like to introduce our second speaker for today, Puan Amelia Azman. She is the clinical scientist in Molecular Diagnostic Unit under Specialized Diagnostic Center at IMR. She holds a master's degree in biotechnology, specializing in population genetics from University of Malaya, Kuala Lumpur. Her research area of interest are molecular genetics of rare genetic disorders and population study of genetic syndromes. She has presented some of her work at national and international conferences on rare genetic disorders such as Prader-Willi syndrome and myotonic dystrophy type 1. She is involved with developing, optimizing, and troubleshooting of numerous molecular diagnostic tests for the detection of inborn errors of metabolism, mitochondrial and neurogenetic disorders, as well as other genetic syndromes in the unit. It is our great privilege to have Amelia with us to share her research. Her presentation title is Prader-Willi and Angelman syndromes, establishing diagnosis using molecular genetic testing. Over to you, Amelia. The screen is yours.
Thank you, moderator, for the very kind introduction. Hi, everyone. My name is Amelia. I'm from the Molecular Diagnostics Unit. Thank you for joining us today. Just now, we already learned about CDG. So now, I would like to introduce another two disorders, which are Prader-Willi and Angelman syndromes. My topic for today is Prader-Willi and Angelman syndrome, establishing the diagnosis using molecular genetic testing. So maybe some of you are familiar with what Prader-Willi and Angelman syndromes are, but maybe some of you have not even heard of PWAS before. So not to worry, I will briefly explain what PWAS are and what causes PWAS, and then we'll move on to the methods and the results. And lastly, we'll discuss on the importance of using molecular genetic testing to establish the diagnosis of PWAS. Okay, Prader-Willi and Angelman syndrome, or PWAS, are neurogenetic disorders, and they are also genomic imprinting disorders with the prevalence of 1 in 15,000 live births. So these two disorders are often discussed together, but they are actually two distinct disorders. So first, we will talk about Prader-Willi syndrome, or PWS. The mortality rate for PWS is 3% per year, which is slightly higher than the general population. And the causes of death in children are respiratory illnesses, dysregulation of temperature, while for adults is usually due to obesity-related cardiovascular problems. Some of the common symptoms for PWS are hypertonia in newborns, developmental delay, obesity, short stature, and also hypogonadism. So PWS patients usually they would develop hyperphagia, which is extreme hunger, which will cause them to eat more and constantly, and they will become obese. Now, moving on to Angelman syndrome, these patients usually would have normal life expectancy, and their features are a happy demeanor. So you could see them frequently smiling and laughing. They also have developmental delay, gait, and also seizure. Okay, the reason why Prader-Willi and Angelman syndromes are often discussed together is because the cause of these two disorders lie in the same position, which is in the 15q11 chromosome. So if you look at the image here, you could see these blue boxes, which are paternally expressed genes, and the red boxes, which are maternally expressed genes, while the green boxes are biallelically expressed genes. So Prader-Willi syndrome, PWS, occur due to the absence of the paternal allele here, while Angelman syndrome occurs because of the absence of the maternal allele. So how did these genes become absent could be due to several molecular mechanisms, such as a deletion, a uniparental disomy, an imprinting defect, or an imprinting center deletion. So these four mechanisms could occur in both disorders, PWS and AS, but for AS, there's also two other mechanisms, which are the UBE3A mutation and also because of unknown reasons. So all of these mechanisms have different proportions, and we can see that deletion is the most common mechanism for these two disorders. The proportion is up to 75%. In PWS, imprinting defect is the least common mechanism, while in AS, the least common mechanism is uniparental disomy. It is very important to determine the molecular mechanism because from the molecular mechanism, only then you could tell the risk to SIPs. The risk to SIPs would tell you the recurrent risk. And from here, you can see that the risk could be negligible, which is less than 1%. It could also be up to 50%. So patients with an IC deletion and a UBE3A mutation would have a recurrent risk of 50%. And this data here is also important for genetic counseling. Okay, let's move on to the methods that are used to detect PWAS. So PWAS are genomic imprinting disorders. So the first thing that you want to do is to detect the methylation status at the SNRPN region. And the two methods that could be used to detect the SNRPN, the methylation of the SNRPN, are MS-PCR and MS-MLPA. So these two methods could detect if there's a hypermethylation, which means that the patient has PWS, and if there's hypomethylation, which means the patient has Angelman syndrome. And from here, you can already confirm the diagnosis. However, MS-MLPA is considered a more superior and better method compared to MS-PCR because it could also detect the copy number, which will tell you the molecular cause that would cause the abnormal methylation pattern in the SNRPN region. So some of the molecular causes that could detect are large deletion, IC deletion, uniparental disomy, and imprinting defect. If you are familiar with the European Molecular Quality Network, which is an external quality provider, they have stated that majority of the laboratories in the world actually use MS-MLPA as the first step to detect PWAS. And also, I would like to note that it is the gold standard method for diseases caused by abnormal DNA methylation. So even though MS-MLPA is a very great method, it has its limitations. And one of the limitations is that it could not distinguish between a uniparental disomy, UPD, or an imprinting defect case. So if you are interested in knowing if the patient has UPD or ID, you have to proceed with microsatellite analysis or MSA. Okay, these are the steps to do MS-MLPA. Firstly, we will extract the DNA from whole blood samples from the proband, and then we will carry out MS-MLPA using a kit from MRC Holland. MS-MLPA consists of denaturation, hybridization, ligation, digestion, and then PCR. And then we will carry out fragment analysis. And lastly, we analyze the results using a software called Coffilizer. When you analyze your results using Coffilizer, this is what you'll see. These are the probes that are included in the kit itself. And here, it will tell you the copy number. And next to it, it will tell you the methylation. So when you do your MS-MLPA, there will be a site that will be digested using a restriction enzyme. So this is the NSRPN digested site. So when you want to diagnose your patients, the first thing that you would do is you would focus on the digested site here. So if you see for a normal patient, the patient would have a 50% methylation status. So this is normal. But for Prader-Willi syndrome patients, the patients would have a hypermethylation or 100% methylation status. So as you can see, all of these patients here have about 100% methylation at the SNRPN site. So it means that these patients have Prader-Willi syndrome. After you have figured out the methylation status, you will look at the copy number or the cause of the methylation. So if you look at the normal patient here, the copy number would be about 1. But for patients, like for example, this case, you could see that the copy number is halved. So it means that there has been a deletion that occurred in this probe. Okay, so there are two types of deletions, which are class 1 and class 2 deletions. And class 1 deletion spans from NEPA1 to OCA2, while class 2 deletion spans from MKRN3 to OCA2. And then there's also IC deletion, whereby the deletion happens in the SNRPN region here. So when we get cases like this, we would request for the parental sample to see if the parent also has the same IC deletion. This is important because if the parent has the same IC deletion, the recurrent risk is 50%. Okay, for patients with Prader-Willi syndrome due to UPD/ID, you could see that there's a hypermethylation, but the copy number seems normal, which is 1. So again, as I've mentioned just now, MS-MFA cannot distinguish between a UPD and ID. So if you want to distinguish and tell them apart, you have to proceed with microsatellite analysis. These are the results for Angelman syndrome patients. Instead of a hypermethylation, they have a hypomethylation, where the methylation is about 0%. So again, after you have figured out the methylation status, you will look at the copy number. And again, there could be a class 1, class 2, or IC deletion, as well as could be a UPD or ID case. And if you remember in the earlier slides, I've mentioned that there could be a possibility of a UBE3A mutation for Angelman syndrome patients. So if you get a normal result, normal methylation pattern and normal copy number, you will proceed with UBE3A sequencing. So the possible outcome would be no pathogenic variant, a point variant, or point mutation, or a deletion like this case. So this patient has a deletion in the exon 17 of UBE3A gene, and this variant has not been reported in the human gene mutation database. Thus, it is a novel variant. And when we check the pathogenicity prediction using VASM, it was found to be pathogenic. Okay, I've mentioned MSA a few times before. MSA actually stands for microsatellite analysis. It is also the same method that is used for DNA profiling of criminals, like what you see in crime shows. It is also the same method used for paternity testing. So for us, we're using MSA to look at the inheritance of the alleles. So it is also the gold standard for uniparental testing. The first step that we do is to extract the DNA from the proband and also the parental samples. We use whole blood samples, and then we'll proceed with PCR using microsatellite markers, fragment analysis, and then lastly analyze the results using a gene marker software. Okay, so before you proceed to MSA again, of course, you have to do your MS-MRPA first and then detect the methylation status in the patient. So if 100% methylation, the patient has PWS, 0% methylation, the patient has AS. Okay, so you will expect to see these two results when you run your MSA. A patient with a UPD will have this kind of inheritance. So the green alleles here are the father's alleles, the red alleles here are the mother's alleles. So the proband or the child, for this case, inherited only alleles from the mother, so only from one parent. It could also be that the proband or the child inherited alleles only from the father. So when you see this kind of case, it is a case for UPD. For ID cases, the proband would inherit one allele from father and one allele from mother. So this is how you distinguish a UPD and ID using MSA. This is our real patient's result. If you can see here, there are 9 loci or 9 markers situated in the 15q11 region. Okay, so what we would do is you would look at the inheritance of the alleles. Is it maternal, paternal, or by parental inheritance? So after you analyze all of the 9 loci, we have discovered that 5 out of 9 of the loci were maternally inherited. So some papers have published that you only need 2 informative loci to tell a UPD and ID apart. So for this case, we have 5 informative loci. So we can confidently say that this patient has Prader-Willi syndrome due to uniparental disomy. So the risk to SIPS is less than 1%. This is another example. Again, you have 9 loci, and you analyze all the different loci. And after we analyze the results, we found that this patient had biparental inheritance in all of the loci, which means that this patient has Prader-Willi syndrome due to imprinting defect. So the risk to SIPS is less than 1%. Okay, I think by now people have understood the importance of understanding the molecular mechanism because it could tell you the recurrent risk, which is important for genetic counseling. But there's also this thing called the genotype to phenotype correlations. So scientists have found that patients with a large deletion in both Prader-Willi syndrome and Angelman syndrome, they have severe phenotypes compared to the patients with other molecular mechanisms. So for example, let's look at Prader-Willi syndrome first. So all the patients with, regardless of the molecular mechanism, would have hypotonia, hypogonadism, and also hyperphagia. But patients with a large deletion would have a more distinct Prader-Willi syndrome facial features and also a higher hypopigmentation. So it's the same for Angelman syndrome patients. Their phenotype would be even more severe. So they would have severe seizures, severe intellectual disability, and also severe hypopigmentation. Okay, for Prader-Willi syndrome patients with UPD, it has been reported that they have higher incidences of psychosis and autism spectrum disorder. So this is not very common for Prader-Willi syndrome patients with other molecular mechanisms. And interestingly, for Angelman syndrome patients with UBE3A mutation, their phenotype is somewhere in the middle, where they could be similar to deletion patients in terms of seizures, absence of speech, and microcephaly, but they are also similar to UPD and ID patients, whereby they have higher development of motor skills. So I feel that understanding the molecular mechanism could help the doctors understand why perhaps patient 1 has this kind of symptoms and patient 2 has this kind of symptoms. So the different symptoms could be because of the different molecular mechanisms. So our unit has been offering Prader-Willi Angelman syndrome diagnostic services since 2009 and up until now. And so far, we've had 2,790 samples, whereby 10.4% of the samples are Prader-Willi syndrome positive and 4.4% of the samples are Angelman syndrome positive. So the higher incidence of Prader-Willi syndrome compared to Angelman syndrome has also been reported in China. Since 2021, we started to offer microsatellite analysis, and so far we've had analyzed 20 cases, whereby 19 of the cases were patients with Prader-Willi syndrome due to uniparental disomy, and only one of the case was a patient with Prader-Willi syndrome due to imprinting defect. So this is also similar as what has been reported, whereby they said imprinting defect is the least common molecular mechanism for Prader-Willi syndrome. I think by now everyone have understood the importance of molecular diagnostics. It is only an accurate way to get an accurate diagnosis. So a lot of doctors here, they would know that a lot of genetic syndromes have overlapping phenotypes like hypertonia and also developmental delay. And the only way for you to pinpoint and get an accurate diagnosis is through molecular diagnostics. So an accurate diagnostics is also important for the doctors to plan the right management and treatment of manifestations for the patients. And lastly, of course, the knowledge that you get from molecular diagnostics work will also help in genetic counseling. So before I end my presentation, I would like to thank all of these amazing staff for their hard work for PWAS diagnostic services in Malaysia. So they are the real experts on PWAS. I'm just the presenter for today. And not to forget, thank you to the other UMD lab members. So that's all from me. Thank you for your kind attention. Thank you. So I'll pass back the floor to the moderator.
Thank you, Amelia, for the insightful presentations. Okay, we will now be moving to question and answer session. Please do not hesitate to send us your questions, and I will try my best to connect you to our distinguished speakers through the posted questions. However, if time does not allow us to address all the posted questions, our speakers will try their best to answer them through email. Okay, it seems that we have received a few questions from our audience. Question number 1 is for Dr. Izzati. Why is it important to characterize each subtype of CDG?
Why is it important to characterize each subtype for CDG? It is because currently CDG have 150 different subtypes, and this subtype it can affect the treatment for the patient. For example, if we know the subtype, we can treat individually. For example, CDG and CDG type 1A. CDG type 1A, this subtype is caused by a deficiency in the enzyme of mutase 2, and it can cause developmental delay, seizure, and other health problems. So if we know the subtype, we can treat, for example, we can treat by changing the dietary. It may include the high protein, low carbohydrate diet, as well as we can uh, we can introduce a speech therapy to address the developmental delay. And another example for CDG type 1B. CDG type 1B is because of the deficiency in the isomerase. So the deficiency of the isomerase we can treat by giving the regular infusion of the sugar called mannose, and also we can treat using antibiotic because CDG type 1B can cause a recurrent infection. So to overcome the problem, if we know the specific subtype, we can give a proper treatment to the patient. That's
Okay, thank you, Dr. Izzati, for the answers. Another question from you, from Nurul. Why do you choose Multitolf over IEF and CZE?
Okay, why Multitolf? As I said before in my presentation, there are lots of benefits on Multitolf technique. Before this, we are using IEF and also CZE, and there are several disadvantages. For example, it cannot provide a subtype information on CDG. It is less specific because it can detect polymorphism, variant, and also interference. But in Multitolf, the advantages is that it is more sensitive. It has high throughput. We study the glycan pattern, and the glycan pattern is more sensitive and more specific. And the best characteristic is that it requires very low sample volume, and it is important for the PITS patient. To give a very low sample volume. That's it.
Okay, thank you, Dr. Izzati. Now, the next question is for Amelia from Sarah. Is prenatal testing possible for Prader-Willi and Angelman syndrome?
Okay, thank you for the question. Prenatal testing is possible for both methods that we use in the lab, which are MS-MLPA and also MSA. But we require the DNA to be extracted from either the amniotic fluid, the chorionic villus sampling, or the fetal blood. And please make sure when you send your samples, it has to be free from DNA maternal contamination. We don't extract the samples ourselves because we don't have the kit to extract samples from amniotic fluid or the fetal blood. So, but we can accept your DNA samples. We can run the test for you. So yes, we can run prenatal testing for PWAS in the unit.
Okay, thank you, Amelia. So another question to Dr. Izzati for CDG. Did you get many samples screened for CDG from Peach Moon YouTube?
On average, we are receive 8 sample monthly for CDG at our unit.
Okay, another question. What is the mortality rate for CDG?
Mortality rate. Actually, CDG, mortality rate for CDG is depending on the specific type and also the severity of the disorder. So in some type of CDG, it may affect the essential glycosylation pathway, and when it affect the essential glycosylation pathway, it can cause severe and fatal symptom in infancy or early childhood. But other CDG subtype may have milder or later onset symptom. For example, the common PMM2 CDG. It is estimated that 20%, the mortality rate is up to 20%. It is due to, in infancy, due to the several multisystemic involvement such as liver, neurology impairment, and coagulopathy. And in some other subtype, for example, CDG type 2A, they are less severe form of the CDG. So, it is important to take note that CDG is very unique and the prognosis and the mortality rate is depend on the various factor and depend on the subtype of the CDG itself.
Okay, we have another one for Dr. Izzati. Multitop has high throughput, but did you get many samples for CDG, and is it worth the cost of the equipment and reagent? From Hamizah IMR.
As I said before, on average, we receive about 8 sample monthly. So the cost it is comparable to the IEF and also to the CZE method for now.
Okay, now we move on to Amelia. Question from Dr. Hamizah MR. Instead of molecular confirmation test, what is the screening test for Prader-Willi and Angelman syndrome?
As far as I know, you can do cytogenetic testing. If I'm not mistaken, HTA or Hospital Tunku Azizah do provide that service. So I think what they can detect from fish or karyotyping work is that if they can see there's a deletion in the 15 chromosome, there could be some problems that would lead to Prader-Willi or Angelman syndrome. So I guess if you have done your cytogenetics work, you can do your confirmation at our unit where we can run the MSMRPA and the MSA for you.
Okay, Mila, we have another one. If the results are negative for Prader-Willi Angelman syndrome, what test should I do next? From Dr. Kit Meng.
Okay, I think not necessarily that you will get positive results when you send your samples, right? It is because the symptoms for genetic syndromes are very confusing and they overlap with each other. So I think maybe the first step that you could do is to discuss your case with maybe again a neurologist or a clinical geneticist and get their insights on what the patient might be having. Other than that, you could try to go for next generation sequencing. There are gene panels that could detect, for example, for syndromic obesity or for neurogenetic disorders or for developmental delays. You can go for that, or you can also go for whole exome sequencing. You probably will get your answers from there, or if you still can't get your answers from WES, you can go for whole genome sequencing. So I think that's the only way for you to try to find and pinpoint what kind of disorders that the patient would have.
Okay, I think we only can say one more question, and this is to Amelia. What are the treatments that are available for Prader-Willi Angelman syndrome?
There is actually no cure for genetic disorders. There's what the doctors can do are actually just to manage and treat the manifestations. So for example, for Angelman syndrome patients with seizures, the doctors can give them anti-seizure medications. And also if they have developmental delay, the patients could go for a occupational and also physical as well as speech therapy. While for Prader-Willi syndrome patients with short stature, they could be given growth hormones. And if they have hypogonadism, they could be given sex hormones. If they have feeding difficulties when they are newborns, they can be given special nipples to help with feeding. And lastly, perhaps if they also have language developmental delay, they could also go for speech therapy.
Okay, I'm afraid that it's all the time that we have for questions from participants. Any additional questions will be answered by our speakers through email. We hope that this webinar has shed light on some research work that we are conducting at our institute and would spark interest and initiate research collaboration with others. Ladies and gentlemen, now we have come to the end of the webinar session. I would like to thank all participants for their support to make this session successful. You may subscribe to our National Institute of Health YouTube channel and click for notifications whenever there is a new post. Before I conclude, I would like to announce that we will have the next session IMR-SEAMEO webinar series on the 31st March 2023, so do mark your calendars. The theme for the next webinar is Zebrafish Model, a glimmer of hope from HMRC team. We hope you can join us at the next session. Until then, many thanks from all of us at IMR. Take care and stay safe.